Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Today I want to show you how I perform a bacteria enumeration lab simulation. So, what is mean by enumeration of bacteria? Enumeration of bacteria is part of daily routine. The lab microbiologist often has to determine the number of bacteria in a given sample as well as having to compare the amount of bacterial growth under various conditions. Enumeration of microorganisms is especially important in dairy microbiology, food microbiology, and water microbiology. Knowing the bacterial count in drinking water, fresh milk, buttermilk, yogurt can be useful in many aspects of industrial microbiology. Bacteria are so small and numerous. Counting them directly can be very difficult. Some of the methods used involve diluting the sample to a point at which the number of bacteria has been reduced to very small numbers. This enables an estimate to be established for quantifying the bacteria. Direct counts of bacteria require a dye to be introduced to the population of bacteria to allow the observer to view the bacteria. Next. So for today, I want to show you how I perform viable plate count method. So this is the definition about viable plate count, which is one of the most common methods for enumeration of bacteria. Serial dilutions of bacteria are plated onto an agar plate. Dilution procedure influences overall counting process. The suspension is spread over the surface of growth medium. The plates are incubated so that colonies are formed. Multiplication of a bacterium on solid media results in the formation of a macroscopic colony visible to naked eye. Next. So this is the more description about this method. This is also the same. Next. CFU per milliliter of sample equal with the number of colonies times with the dilution factor of the plate counted. So this is the procedure of viable plate count and have this procedure have four sample include water from a drinking fountain, boil water from a drinking fountain. Third, water from the local river. And lastly, boil water from the local river. So next, this is the procedures. First, take 6 dilution tubes, each containing 9 milliliter of sterile saline. Second, dilute 1 milliliter of a sample by withdrawing 1 milliliter of the sample and dispensing this 1 milliliter into the first dilution tube. Third, using the same procedure, withdraw 1 milliliter from the first dilution tube and dispense into the second dilution tube. Subsequently, withdraw 1 milliliter from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is completed. Fourth, transfer 1 milliliter from each of only the last 3 dilution tubes onto the surface of the corresponding agar plates. Fifth, incubate the agar plates at 37 degrees Celsius for 48 hours. And lastly, choose a plate that appears to have between 30 and 300 colonies. So, 7. Count the exact number of colonies on that plate. And lastly, calculate the number of CFU per milliliter of original sample as follows: CFU per milliliter of sample equal to the number of colonies times with the dilution factor of the plate counted. So first click on the dilution tube rack of test tubes to bring them to the table. Each of dilution tubes contain 9 ml of sterile saline solution. So next click on the water samples to bring the samples to the table. Now click on the eyedroppers to withdraw 1 milliliter of sample number 1, which is fountain water, and dispense this to the first dilution tube. So next, click again on the eyedropper to withdraw 1 milliliter from the first dilution tube and dispense into the second dilution tube, and subsequently withdraw 1 milliliter from the second dilution tube and dispense into the third dilution tube. Continue doing this from tube to tube until the dilution is completed through dilution tube number 6. Okay, click on next. Okay, the dilution for each of the 6 dilutions tube can be summarized in the image below. So this one, next. So next, we will be inoculating agar plates with the last 3 broth culture dilutions. Click on the agar plates on the shelf to bring them to the table. Now click on the eyedroppers to transfer 1 milliliter of dilution number 4, sample number 4, to plate number 1, 1 milliliter of dilution 5 to plate number 2, and 1 milliliter of dilution 6 to plate number 3. So eyedroppers, dilution number 4 to plate number 1, 1 ml of dilution number 4 to plate number 2, and 1 ml of dilution number 6 to plate number 3. Okay, done. Next, click on the pencil to label agar plates number 1 with a dilution factor 10,000, plate number 2 with a dilution factor 100,000, and plate number 3 with a dilution factor of 1 million. Okay, done. Next. Click on the agar plates to place them in the incubator at 37 degrees Celsius for 48 hours. And click on next. Okay, click on the incubator to bring all of the inoculated agar plates to the table. Each of the groups of inoculated plates is labeled with the source of their respective samples. So for the first one is the fountain water. Okay, this one 10,000. So you see that there is the number of bacteria. Okay, for the 100,000. Alright, this one. And this is 1 million. The number of bacteria was decreased. So for the second plates, which is boil fountain water. So for the 10,000 plates, so there is the bacteria, the many of bacteria. So this is the 100,000. The number of bacteria slowly decrease. And lastly, 1 million. There is no bacterial found. So next for the third plates, which is river water. For the first plate, 10,000. There is many of bacteria count. This is 100,000. And this is lastly, which is 1 million plates. So this is the number of bacteria. And lastly is the boil river water. This is 10,000 plates. Okay, this is the number of bacteria. And this is the 100,000. And lastly, 1 million plates, which is this is the count of bacteria. So that's all. That's all for today lab simulation. Thank you.